theory of inheritance, and we now know what a gene is in a better sense because we know the molecular basis of it. But he really was forward thinking in that realm, and he suggested that if you inherit particular genes from your parents, whether you're a pea plant or a person, you will end up with a statistical relationship based on what the phenotype of your parents were. And he basically came up with the idea that if genes were separated on different chromosomes, that when you looked at the possible gametes that could be produced when you had a heterozygote for both A and B, that you would get 25% of the progeny having each of those individual phenotypes. And if you look through Gregor Mendel's actual notes, and some of us have actually gone back and looked through the Library of Congress's scannings of all his notes and everything like that, Gregor Mendel was right about things about 1/3 of the time. And it happens that what he told everybody about was flower color, color of the seeds, whether they were wrinkly or not. And these happen to be genes that were, these happen to be phenotypes that were generally controlled by one gene, and those genes independently segregated from each other, typically because they were on different chromosomes or extremely far apart from each other on one chromosome. And that gave this independent assortment of homologs, and that works if they're on different chromosomes or if they're extremely far away from each other on one chromosome, such that they're effectively separated independently of each other. However, if you read about 2/3 of Gregor Mendel's notes, you'll see that his 25, 25, 25, 25, it just didn't work, right? It was no good at all. And he just didn't tell anybody about that bit. He wrote it down, and that was fine, but what he realized is that under certain circumstances, there were genes that didn't necessarily independently segregate from themselves. And that would make sense if the genes were on the same chromosome. If you have crossovers then between the homologs in a bivalent, you can influence the co-transfer of individual phenotypes into the haploid gametes. And so you basically end up with some small percentage X where a crossover happens between A and B. Now, if you're looking at a whole population of peas, and every individual pea has some probability of having that crossover happen, the greater the distance between them, the more likely they are to have a crossover between them. The closer they are together, the more likely they're going to co-segregate with each other. And basically, Mendel found that his stuff didn't work. So Mendel decided to ignore it. That was fine. He was still remembered. Then there came a really humble guy, and you realize how humble he is in a second, named Thomas Hunt Morgan, and he came about 150 years later. And he was one of the nerds who went back and looked at Mendel's stuff and said, Mendel was right about 1/3 of the time, but he was wrong about 2/3 of the time. So there needs to be something else about this. And I've yet to find out why this guy really liked fruit flies. People like a lot of weird things on the planet, but I've never really met anyone who was like, wow, fruit flies. I really need to deal with them. But they're kind of convenient to use in the lab. Pea plants, you plant them in the spring, they grow up, you might get 2 crops in, then you're a monk the rest of the year. So you can't do lots of fancy genetics with them. Fruit flies, you just put a banana out and they're happy. And they breed all year long. Anyone who's ever got them in their kitchen knows how much of a pain they are to get rid of. And he decided that fruit flies had lots of body parts and were really easy phenotypes to score on. And they were big enough to see without, microscopic intervention. You could keep them in the lab, and you could basically just feed them like a peanut butter-like material, and lots of them would develop in there. And they had interesting larval phenotypes and interesting adult phenotypes, and you could mess with them. So Thomas Hunt Morgan decided that these guys, and it was known about the time that he started dealing with it, there was really only 4 chromosomes in there, and that he could do a lot of things with them. And so what he decided to do was use something that he called a linkage analysis. How often do 2 genes actually go with each other, and how often do they not? So if we didn't have chiasma, we didn't have crossovers in a bivalent, then capital A would always go with capital B, and lowercase a would always go with lowercase b. And those just represent 2 different alleles of 2 different genes, right? Nothing exciting. But Mendel even noticed, and other people have noticed before, that under some conditions, there was actually a crossover between the homologs, and then you would get this small percentage x. And the amount of x is a reflection of how close they are on the chromosome. The closer they are together, the lower the probability is that there would be a crossover event separating the two between them. And you can actually more or less determine the distance between A and B in some sort of arbitrary units if you knew that you scored a large number of probabilities. You can do that in peas, but it takes a long time. In fruit flies, one has white eyes, and one has red eyes, and one has antenna where its feet should be, and one has regular feet where its feet should be. You can sort of score those two phenotypes going together. And so he came up with this really neat term, a centimorgan. Now, remember what the guy's name was, right? Thomas Hunt Morgan. So he named it after himself. Centi means 1 out of 100, right? You have centimeters, right? It's 1/100th of a meter. So for every percent of recombination frequency, Thomas Hunt Morgan decided that that should be a unit named after him, and it should be a centimorgan. So you should never forget what it actually is. And so he put that into the literature. It's more or less stuck, because what else are we going to call it? We could call it like centi-somethings, but we might as well call it after the guy who first came up with it, give him a little bit of praise. The fact that he named it himself sort of annoys me, but we'll go with that. So you can pretty much figure on your exam, there's going to be a question that says something like that. And you can't figure that out, I'm sorry, you deserve to fail. Okay? It's just, what's the distance in centimorgans? Geez, it's the number 3. Okay, let's go with that. You would be surprised how many people get that wrong on the exam, but we'll go with it.